So good morning. Thank you all for joining us this morning for February's ECAP Recap. It's our webinar series hosted by the ECAP team, the Environmental Compliance Assistance Program. My name is Stacy Gadd. I am our Outreach and Education Scientist on the team. Today we're going to be talking a little bit with Jacob Culp from the Division of Water, and he's going to be talking about how his section monitors the waters of Kentucky, make sure the health of everything's going okay in our waterways. But before we get started in that, I just want to do a little bit of a little bit of webinar housekeeping and just do a brief intro on ECAP and what we do. So webinar housekeeping, today's webinar is going to be about an hour. I'm going to do a very brief introduction here. We're going to ask that you attendees stay on listen only mode for right now, but we will have a recording of this available here in a couple of days. Just once we get it all uploaded to YouTube, I'll send everybody a link via email. All right, so the Environmental Compliance Assistance Program. Our main goals are to help businesses understand and comply with Kentucky's environmental requirements. Specifically, small businesses, we have a pretty specific definition of that. But if you want more information, please give us a call, give us an email. I just want everybody to know that we are here to help. Kentucky's ECAP team is kind of unique in that we do some other things. We also help answer the environmental helpline and kind of direct members of the public, members of businesses to the different divisions to get assistance and answers to their environmental questions. So we function as Kentucky's Small Business Environmental Assistance Program, which is a requirement of the Clean Air Act. And we assist small businesses that are specifically less than 100 employees, independently owned and operated, and not a major source of air emissions, since this is funded under the Clean Air Act. What do? We can provide small businesses with detailed assistance on their air permits and applications and different record keeping items such as rolling total spreadsheets. We can offer general multimedia technical assistance to members of the public, which just means we also answer questions about air, water, and waste, about water and waste, as well as air, which is not a requirement, but Kentucky has chosen to make our SBEEP multimedia. So we also can help develop educational documents. We have different fact sheets and stuff available on our website. We have some recorded online modules, and we hold webinars like today and some in-person training like our ACC SAMR workshop that we did back in January. So what we don't do, we do not, it's important to know what we can't do. So we don't issue notices of violation. We are not a regulatory entity. We are just here to help. So if you employ our services, then we have a legal, like a discretion, so that we cannot report anything we see. Even if we do see a violation, we have to give you time to amend that and remediate any issues before we report anything to like the Division of Air Quality. So we can't make determinations in any cases. We can make recommendations for how to remediate things. We can't interfere with inspector reports or findings, and we can't be a sole corrective action that is required in an NOV. We're just here to help make those changes. So our enforcement discretion means that you guys are protected. Any small businesses are protected whenever we come to look at a site, if we do a site visit. Upcoming events, we have another ECAP recap coming up March 4th on voluntary environmental audits here in Kentucky. And then sometime in April, we're not entirely sure the date yet, we're going to do an ECAP recap on data centers. And then you can find more information on this stuff at the website that we've got linked. And without any further ado, I'll hand this over to Jacob, our environmental biologist supervisor over at the Division of Water. So thanks so much, Jacob.
I'm going to be talking about, as Stacy mentioned, monitoring sections, basically our programs and some of the overview of what we do, which is biological and water chemistry monitoring across the state of Kentucky and our streams, rivers, and lakes. So our monitoring section is in the water quality branch of Division of Water. I'm Jacob Culp, the supervisor. I've been with the monitoring section for 14 years, acting in various project coordination roles, For several years, I coordinated our ambient rivers program and our reference reach program. We have 4 environmental scientist advisors that have anywhere from 11 to 6 years of experience. And we have 4 environmental biologists, a couple of fairly new ones that have been with us for less than a couple years, and then some that have been around for a few years. So typically our section has 9 to 11 people. We also usually have a couple of seasonal interns to help with field work in the summer months. So what does monitoring section do? We monitor and sample statewide. Our big things that I've kind of broken it into different categories here. We monitor for aquatic life. That includes macroinvertebrates and fish. Everywhere we do aquatic life sampling, we also do a rapid habitat assessment. We monitor water chemistry, and we also have a separate program for fish tissue, and this is related to fish consumption, and some of our advisories and assessments related to fish consumption. So I've kind of got that set as a separate piece of our monitoring that I'll cover. So first, our macroinvertebrate sampling. We use a index, which is our macroinvertebrate bioassessment index, and I'll get into the specifics of that a little bit later. So this is a high gradient methodology that we use in streams and rivers. We also have a low gradient methodology that we use in certain regions of the state. And I'll demonstrate that, show that a little bit later. Our high gradient methodology, we use a semi-quantitative method of riffle kicks, and you can see in this photo here, this is one of our staff doing a riffle kick in a stream in Kentucky. And these are quantitative. They're quarter-meter squares. We do 4 of them in different riffles as part of the sampling. And then we also have defined multi-habitat sampling that we combine with those riffle kicks. Our low gradient method is a little bit different and focuses on partitioning habitat to what's available. This is that you see the max 20 tab, so that's kind of assigned specific habitats. Once we're done with our macroinvertebrate collection in the field, those are brought back to the lab, sorted and picked, and all the macroinvertebrates are identified. We've got well-documented lab methods for all of our work as well. See here one of our staff spending some time on the scopes. That's a lot of what our staff does when they're not in the field. They're in the lab doing scope work. So the MBI, which our index that we use for our macroinvertebrates, once we're done with our sampling and our lab ID, we assign a score with this MBI, and that is what we use to assess the streams. The score has several different metrics in it that make up the total index. The metrics are genus taxa richness, EPT richness, and EPTs are each families of more sensitive organisms, mayflies, stoneflies, and caddisflies. We also have a modified index, which is meant to discriminate organisms that are intolerant to any kind of organic enrichment. We have a modified percentage, which is relative abundance of EPTs, and this sort of discriminates to remove any pollution tolerant organisms like caddisflies. We have a percent Ephemeroptera category, which is again a relative abundance of the total number we collected. And these organisms are sensitive to pollution. And then we have a relative abundance with chiros and oligochaetes, which are worms and are tolerant to pollution. And finally, we have a relative abundance, a percentage of primary clingers, which gives us a better idea of what the habitat is like in a stream. They're responding to sedimentation and siltation, so that's generally a good way to determine what the habitat is like based on a macroinvertebrates, what's living there. So this is related to substrate stability. the bottom here, you can see a variety of organisms. These are on the far ends. You've got mayflies and stoneflies. One of them wild there on a rock in the stream. The other two are preserved and taken from under a scope. So as I mentioned, we have bioregions that we use for our macroinvertebrate MBI, and some of this is related to our methodology that we do in the streams as well. For everywhere in the state except for the MVIR, the Mississippi Valley Interior Region, in the brown on the map, we use a high gradient methodology, and that's the 4 quantitative riffle kicks I was talking about earlier. That method is used across the state in the mountains, in the bluegrass, and in the pennyroyal. In the MVR, we have a separate method, and that's that max 20 jab methodology, which is driven based on habitat. There aren't really low gradient streams in the western part of the state, and so that methodology was developed to work around streams that are low flow without riffles. When we look at the MBI too, where our index, it is bioregion specific. So a score in the mountains is sort of naturally, not naturally, but it's designed so that various bioregions of the state are categorized to meet what the bioregion has to offer as far as stream quality. This next slide is just kind of what the ratings look like, and you can see the BG is bluegrass, MT is mountains, PRs pennyroyal, and then you've got your Mississippi Valley on the far right. They're broken up into wadable and headwater streams. Our headwater streams are anything that drain less than 5 square miles, and wadable streams are anything from 5 square miles up to 200 square mile catchment areas where our macroinvertebrate methodology is not super effective, anything over 200 square miles. So you can see, for example, an excellent score for an index in the bluegrass would be anything greater than 70 in a wadable stream, while in the mountains that bar is set a little higher at 82. And then in the MVR, where we know streams are more impacted, that bar is lower, where anything greater than or equal to 58. You can see similar for headwater. Depending on the bioregion, you've got various scores for excellent, good, fair, poor, and very poor, which would be what we call a narrative rating of the stream condition, and that is based fully on the macroinvertebrate sample. So along with our MBI ratings, we also have a fish index that we use to give a score for all of our fish across the state. And this is called the Kentucky Index of Biotic Integrity. Unlike the macroinvertebrate sample, the methods are the same for all bioregions. There are no differences in the methods for bioregions. We use backpack electrofishing or barge electrofishing. In this upper right picture, you can see our staff out doing barge electrofishing. And so the way electrofishing works is we're actually putting an electrical current in the water, shocking the fish, basically stunning them. You know, the goal is not to kill them, just to stun them so that we can collect them and ID them. And the backpack is basically a version of the barge that is run by a battery, puts an electrical current into the water, shocks the fish. We're able to collect them, identify them, and record that. So for backpack or barge electrofishing, we have set times for our methods. For headwater streams, again, anything less than 5 square miles, or wadable streams, anything greater than 5 miles up to 200 square miles. We also have a seine component of our index methods that is very habitat specific. So anytime there are areas that are deeper with slower moving water pools, we seine those habitats. for 30 minutes. So again, with our KIBI metrics, our index for fish, after sampling and any sort of lab ID that is done, we use these KIBI scores to assess streams. The metrics associated with these are native taxa richness. We use these for weightable streams. Again, anything over 5 square miles. We have a richness in metric for our darter species, mad toms, and sculpins. We have a metric for any intolerant species richness. The intolerant species are basically designated as sensitive to any pollution. We also have another metric that is simple lithophilic spawning species, which is just the number of individuals that use simple spawning, just sort of open spawning. And often this is another good habitat sensitive method. We can tell a lot about sedimentation, siltation, habitat driven features based on the number of simple lithophilic species that are present and that total abundance. We have a percent relative abundance insectivores and a percent tolerant species. These would be species tolerant to pollution. And then just for our headwater streams smaller than 5 square miles, we have a percent facultative headwater. These are species that would basically not normally or naturally be present in a small headwater stream, but because of changes to habitat, they're found there due to disturbances. So that's a sort of a separate category of percentage of species that would normally not be found in headwater. So again, these are our fish bioregions. You'll notice that there's a couple of additional bioregions specific to fish. A lot of this is based on unique fish evolutionary biology. Of course, fish are not able to move across borders as well as macroinvertebrates can. So often you'll notice Cumberland above the falls. So the waterfalls at Cumberland Falls, anything above that presents as a separate unique bioregion in the state for fish. So we have a separate bioregion there above the falls. And then we have a separate bioregion in the upper Green and Pennyroyal part of the state. Other than that, these bioregions are very similar. They're actually exactly the same as our macroinvertebrate index bioregions. And with the KIBI, our fish index, you'll see we have these, again, these unique categories on the far left. You'll see that's the Cumberland above the falls. We have bluegrass. We also have the Green River, which is specific to fish, our mountains, our Pennyroyal, and then our Mississippi Valley interior region as well. There is no weightable and headwater separation here. Instead, these are driven by these unique bioregions, Cumberland above the falls and Green. You'll notice again the bar is set differently for different parts of the state. In the Green, a score of excellent is harder to achieve than it is in the bluegrass. And a lot of this is based on our reference data that went into developing these indexes, which I'll get into a little bit later on. So again, the same classifications for a narrative rating for a stream. We have excellent, good, fair, poor, and very poor. So finally, when we do any of these biological assessments at a stream, we're also doing a habitat assessment. We have a separate habitat assessment for any high gradient stream and any low gradient stream. And these low gradient streams are just the ones in the Mississippi Valley interior region. For the high gradient and low gradient, we have several factors that overlap that we score. We have epifaunal substrate, available cover, and this is focused on what sort of cover and substrate is available for usage by any aquatic life. We have sediment deposition, channel flow status, any channel alteration, bank stability, vegetative protection on the bank, and then the riparian zone width. And this is basically just how wide the forest cover is on either side of the stream. both the bank vegetation protection and the riparian zone width, get a score for each bank. Sometimes one bank may be right next to a road and the other bank be all forest. A good example in this picture on the right is a lot of times in the mountains, we see streams that run right next to the road. The road might be 20 feet away from the stream and the other side is all forest. For our high gradient, there's a few different categories that are not in the low gradient. These are embeddedness. That relates to siltation, sedimentation, that basically would be clogging up any habitat. And that sort of gets its own unique score. And then velocity depth regime, this is the number of kind of unique stream flows and depths throughout. So pools, riffles, runs, do we kind of have all the habitats available possible? And then the frequency of riffles is a separate factor. And the riffles are kind of that fast moving, shallow water is a very important habitat for macroinvertebrates and fish. And our low gradient streams, see David here next to a low gradient stream in Western Kentucky. We have some different characters that we use: pool substrate characterization, pool variability, and channel sinuosity, basically how much curvature and S-shaped curvature does the stream have. So like I said, at each stream we do, we're doing a what's called a rapid habitat assessment. So these habitat assessments then again are categorized by different bioregions and get an overall score of good, fair, and poor. And those are separated again into headwater in the bluegrass and wadable. Everywhere else, the score stays the same for good, fair, poor, whether or not it's headwater less than 5 square miles or wadable greater than 5 square miles up to 200 square miles. So I just threw an example in here to give you guys an idea of what there are normally 10. I just kind of pulled the first 5 and pulled this out of an actual, a real example of a stream. And I've got pictures on the stream at the bottom there. So you can get an idea of how we're grading these things. Our habitat assessments are always done as a team. So it's a consensus that's meant to be fairly, I guess, kind of a consensus-driven score. You can see we kind of have different categories, these condition categories of optimal, suboptimal, marginal, and poor. And we go through as a team and give it an overall score, which relates back to it being optimal, suboptimal, marginal, or poor. You can see here, this is actually one of our higher quality Western Kentucky streams. Overall, it got optimal to suboptimal scoring. Each one of these optimal, suboptimal for every category kind of has a description that we're viewing in the field and making our best estimates to describe the habitat we're seeing. So secondly, besides our biological sampling, water chemistry sampling is the other really big thing that we do as a section. Everywhere we go, every biological sample we take, we have a suite of 4 bottles that we collected, again, for every program. We have a bulk bottle, which is our chlorides, sulfates, turbidity. It includes total suspended solids. We have a nutrients bottle, and this is phosphorus, nitrogen, nitrates, nitrites. We also have a metals bottle that we have close to 20 different metals that we sample for. Some common ones that we have water quality standards for are iron, lead, and copper. And we also finally have an alkalinity bottle. This is basically hardness of the water or acidity of the water and helps determine how impactful the metals are to aquatic life. So again, those 4 bottles are collected everywhere we go. On the right there, you can see one of our staff collecting a bottle right below a riffle. For some programs, we also focus on pesticides and herbicides. And we have a separate program where we focus on E. coli bacteria. And I've got a couple of slides where I'll describe this in a little more detail. We finally have a response program that we respond and collect water chemistry to determine any harmful algal bloom impact. Again, at every site we go to, we have multi-parameter meters, YSIs or XOs. In the picture here, you can see a string of them for some of our lakes work. These multi-parameter probes collect temperature, DO percentage and milligrams per liter, pH, and specific conductivity, which is basically the number of ions present in the water. So we do these multi-parameter meters. They're placed in the streams everywhere we go when we record these physicochemical parameters at every site. So I mentioned E. coli. I'm sure everybody's heard of E. coli in one form or another. We collect E. coli for our ambient rivers program. We also have another section in the branch, the intensive surveys and wetlands. They do a lot of E. coli. We have one program where we focus on it. When we collect these E. coli samples, we bring them back to the field and take them to our microbiology lab. They're basically poured into a standard 100 milliliter jar. We add food for the bacteria that would be present there at that time, and then we incubate those samples for 24 hours to 28 hours. You can see on the right there, anything that is yellow is basically a square that we would count that shows the presence of a fecal coliform bacteria. Anything that's glowing, that's actually glowing from a black light, is fluorescing due to the food that we feed them, with the food that we add to the bacteria sample. And anything that's glowing indicates that there is E. coli bacteria present. So when we come back from the field, we're running these through an incubator with a protein binding food that allows us to tell whether or not, based on color or glow, fluoresce, if we have E. coli present in a site. Of course, the number of squares are recorded, and that gives us a total number of E. coli colonies present at any given water chemistry sample. So finally, I've separated the fish tissue sampling into kind of a unique category for this. Our fish tissue sampling is driven on fish consumption advisories and our fish consumption assessments. So when we're doing our fish tissue sampling, we're using a boat electrofisher. If you guys are familiar with boat electrofishing, this acts a lot like our backpack electrofishing, but we're actually standing on a boat and putting electricity into the water, collecting the fish that we need for our methodology. Because this is focused on fish consumption, we're not collecting small darters or sculpin or anything like that. We're really focused on game fish. So the Centrarchidae is the group of fish that most people know as sunfish and bass. That's our big target by size class, largemouth bass. We target based on size class. For sunfishes, which are not quite as big, or crappie, we do composite samples where we take a set number of samples, multiple fish, combine those into one sample, and do a composite sample. We also focus on bottom feeders, Ictaluridae catfish. The goal there is to try to get at least one catfish from each lake that we sample. We primarily try to focus on channel and blue catfish, as those are more likely to be consumed, but we also will take flathead catfish if that's what we get that day. So once we bring these fish back to the lab, we have a long processing step that we go through. The first step we do is we fillet the fish as anybody would that was going to eat a fish. So we fillet the side of the fish. We use the same left side every time. Once we're done with the fillet, we go through a process of removing all the water. We do this by freeze drying the fillet multiple times. And once all the water is removed from that, we basically turn what was fish tissue, a fillet, into a powdered substance that the lab can take and analyze. We also collect the otoliths from the fish. You can see those there in the bottom left corner. they allow us to get a good indication of how old that fish was. So why are we doing all this stuff? Most of our work is driven on two things: assessments and advisories. So our assessments, we send a report to Congress every 2 years that describes the condition of streams in Kentucky, and we assess new streams as often as we can. Our focuses are aquatic life, both warm water and cold water streams, our contact recreation, which is primary and secondary. Primary is swimming, and secondary would be fishing, kayaking, boating. Contact recreation is mostly driven on our E. coli bacteria. We also have contact recreation on pH. If pH is too low or too high, we would issue a contact advisory, secondary contact assessment for that segment of stream. We have our fish consumption assessments, and all of these have 3 different support categories for assessments: full support, which is green; partial support is yellow on maps, and then non-support red, which makes a lot of sense. And here on the right, I've just pulled from our water health portal, which I'll show on the next slide, an example of what this typically looks like. You can see here in Madison County, Muddy Creek is currently mostly the whole of Muddy Creek is listed as non-support for primary contact recreation because of E. coli data that we've gathered. It is full support for aquatic life, but contact recreation is non-support, and Paint Lick Creek on the western side of the county is full support for warm water aquatic life. So if you're interested in knowing in the county or city you live in what streams have been assessed and what they've been assessed for, our water health portal is a great tool. This is available on the website. You can just Google Kentucky Water Health Portal. This will come up. I've just pulled a quick map here of the kind of home screen. And this gives you a good idea of the water bodies we have and have not assessed across the state too. You can search specifically for a county, a city, or a region, or even a stream or lake, and see what data has been collected and what assessment has been made on that stream. So we also, besides assessments, we issue advisories for fish tissue and harmful algal blooms, which I'll get into. Our fish tissue advisories are done in collaboration with the health department and Kentucky Fish and Wildlife. And all of these guidances, I'm not going to go into super specifics on. I did want to pull so you could see some examples of what these look like. So we have statewide guidance for predatory fish, for bottom feeders, our catfish, and panfish, sunfish, crappie. Typically all other fish we don't have advisories on. In some cases, we have specific lakes that we know have some contaminant issues. And so there's a separate link both on our page and Kentucky Fish and Wildlife page that focuses on specific lakes or rivers that have a specific contaminant in mind, which would be PCB or mercury in most cases. So again, all of our fish tissue data eventually goes into making advisory decisions and assessment decisions that we work with Fish and Wildlife and Kentucky Public Health on. We also do harmful algal bloom advisories. As I mentioned before, these harmful algal blooms are response driven. We don't actually have a program where we're monitoring algal blooms at any given time, but also on our website, we have a report a bloom at feature. So if you do see anything that looks suspect, like any of these pictures above, if you're concerned about a harmful algal bloom, you can follow up and we will respond by going out to take a sample and determine whether or not there is an algal bloom that could be harmful to humans. We work with public health on this a lot too, as they are much more familiar with what the impacts would be to human health. We also have a nice HAB viewer online. Any HABs that have been reported, you can go online and view Again, this is another simple Google search. If you view our website, it'll take you right to it. Finally, the other big thing we use our data for is distribution and species records. We all have collection permits with Kentucky Fish and Wildlife. Anytime we find new records in all of our aquatic life work, those get reported to Kentucky Fish and Wildlife. Any rare species would also go to nature preserves. So now I'm going to get into our program specifically a little bit, and I've got about 20 minutes, so I think I can cover all this. For our monitoring section programs, we have kind of based them in 3 different categories here for this. Our water chemistry focused with no biology, or our ambient programs, ambient rivers and streams and ambient lakes. For our biological sampling, which would be our macroinvertebrate methods index and our fish index, we have a reference trend and special waters program, which would be special waters designated uses, reference reaches, outstanding exceptional waters. Our probabilistic program, which is randomly located by GPS bioassessment. This year, we've added a new program, which is our ambient biology program that ties into our water chemistry program. We also have our fish tissue program under biological sampling. And that is the fish tissue for our assessments and advisories. Finally, our only response-based program is our harmful algal bloom program. So I'm going to go through each one of these and give you a little background on what these programs look like. So our largest program, most data-rich program that we have is our ambient surface water program. The map here, you can see all of the sites associated with the program. There are actually over 175 sites associated with the program. This program is done on a rotating basin. So every 5 years, we rotate through all of these basins you can see in these various colors. This year, I'll describe this year, what we're doing this year next. This program would not actually be possible without our field operations branch. All the field offices assist with the collection of these sites. There's no way our staff could sample these streams all over the state in the amount of times we would need to. So water quality branch, our staff handles the streams within an hour and a half of Frankfurt, and the field offices collect water chemistry at all the other streams. We have 73 primary sites. They're collected bi-monthly every year, and during the rotation, they're collected monthly. We've been doing our ambient surface water program for about 30 years. So there's a lot of data with this program, a lot of water chemistry. We do our 4 major bottles that I've mentioned before, the suite of nutrients, metals, bulk, and alkalinity. We also do pesticides and herbicides at streams where we know there is possibility of impacts from pesticides and herbicides. And during the recreation season, we do E. coli at these. So in 2026, this year, we are rotating through the Green Basin. The ambient surface water program has kind of a weird schedule. It starts in April and goes through March. That's been around for a long time, for over 20 years. I'm not sure how the April to March thing got started, but that's how those program basins rotate. In the Green Basin, we have 16 primary sites that are in green and 26 rotating sites. So in 2026, those sites will be sampled monthly, and everywhere else in the state, all the other green dots in Kentucky will be sampled every other month. Our ambient lakes program, you can see the map here, very similar to the ambient rivers program. We have a rotation. The ambient lakes program, we sample twice a year. We sample in July and September. You can see from the map that our rotation for 2026 is in far western Kentucky. We have a subset of lakes out there that we'll visit both for the rotation and a condition check, which is just kind of an update on a lake that is typically an outstanding state resource water or exceptional use or drinking water facility that provides drinking water. When we do our lakes program, we're out on a boat on the lake. You can see a couple of examples of pictures here. We're doing composite samples with a Kimmerer sample. These are done vertically throughout the photic zone of the lake, and they're done at predetermined depths. Once that composite sample is taken, we do all the same bottles we would normally do, metals, nutrients. We also do pesticides and herbicides. And those are composited from our vertical depths throughout the lake photic zone. We also use our XO logger, which is this big long cable here on the right. And we measure dissolved oxygen and pH until we reach the hypolimnion, where dissolved oxygen cuts off. So we typically have a good profile of all the lakes we go to, where we can look at the dissolved oxygen, the pH, and determine where the cutoff is for oxygen in each lake. At all of these lakes, we also take a chlorophyll A sample, and that actually comes from the composited sample. In 2026, we're doing a special investigation on 3 Army Corps lakes, which we normally do not sample because the Army Corps does their own sampling. We'd like to follow up on these 3 lakes, as we've had some concerns from citizens in these lakes. So this year we're going to be focusing on Cave Run, Green River Lake, and Nolin River Lake. We've got about, across all 3 lakes, we probably have about close to 20 sites that we'll be sampling for our lakes program. We're going to try to cover the scale, the length of each one of these lakes, 2026. So our other program, our big biological program, is our reference reach program. Little quick background on our reference reach program. You can see these in pink here. These are the state's reference reaches. And these streams were actually what created our indexes. These are the streams in Kentucky that are considered least disturbed, and those were used as the baseline for both macroinvertebrate and fish to create the baseline for our reference, for our indexes across the board. So anytime we're collecting a biological sample, we're comparing those to what the highest quality, least disturbed sort of control group of streams in Kentucky. So we make sure when the indexes were created, we make sure that we had least disturbed streams in each bioregion that was the best representation of the least disturbed streams in those areas. So our reference reach program, to follow up on that, for the past decade plus, we've gone to a subset in each bioregion of these streams to determine how accurate our MBI and KIBI is over time, annually, with any seasonal variation, rainfall, any changes in weather conditions for that year. We want to be able to be confident in our indexes that they're consistent. So this reference trend program has been going for over a decade. You can see the sites spread across the state that are visited every year for biology. And this is a quick slide I pulled in from another presentation to give you an idea of what this data looks like when we're collecting this. This is actually the macroinvertebrate index score here. And you can see on the right streams that have consistently scored in the various categories of narrative: excellent, good, fair, poor. Since these are reference streams, we don't have any poor in the mix. And you can see that a subset, about a third of these streams, have consistently been excellent or good. We have a subset in the middle where we do have some variation, and this is where we're interested in determining, like I've circled 2015, whether or not there are factors like weather, rainfall, other seasonal factors that we're not sure about are driving any changes in our metrics or index scores. So this is a long-term program where we're gathering data to look at our confidence in our indexes on streams that we know are high quality. Our probabilistic program is an interesting one. This program, every year we target 50 sites. These sites are randomly chosen using a program that selects a GPS point on a streamline, and we go to that streamline wherever it is. The goal of this program was to give a random, statistically random driven nature to sampling streams. Instead of going to a bridge crossing or an easily accessible point, this program was meant to be completely random within an assessment unit. And every year we're doing 50 of them. This often means we are getting into some weird spots in Kentucky, often private land where we're talking to landowners or places we can't easily access. I've just thrown some pictures in here. We've had places where we've had to access by canoes. You can notice in the center picture that we're all wearing long sleeves and mosquito nets. This was midsummer, and there were mosquitoes everywhere. So we often are on some sort of livestock properties. A lot of Kentucky is farms. So we typically are accessing and communicating with farmers to get permission to access streams. Sometimes we end up behind somebody's house in the mountains. So the probabilistic program takes us all over the state to sample streams in a random place in Kentucky. And this is a quick map or a quick slide up we put together from Attains, which is an EPA website. And this is our data that we have placed into Attains. And this gives you a quick idea of how streams break out based on their macroinvertebrate index score. You can see on the right, you've got all the various categories. And if we do 50 sites a year, what percentage of those streams are excellent, good, fair, poor, and very poor? And you can see from 2017 to 2022, which is as far as we've gotten with the analysis of this, up to 22, that we pretty consistently see the same pattern over and over again, where majority of the streams that we sample are fair, but we do have a nice, kind of curvature to that graph where the mid portion is fair, and then there's not a lot of poor or very poor streams, but we do have a fair number of good and excellent streams that we sample every year statewide randomly. So just a quick graph you can take a look at to see how those index scores look statewide on a random scale. So a few years ago, we also developed a new program where we go back to probabilistic sites that we've been to before to collect more data to make a better assessment. So you can see here we've got monitoring frequency, and this is the number of water chemistry samples we would need to get to determine what is causing any impairment in a stream. So for this year, you can see we've got a dozen or so streams that we'll be visiting for water chemistry based on a biological driven criteria that we want to gather more data for. Our fish tissue program, I've just thrown a quick table up here. These are all the lakes that we'll be sampling for fish tissue in 2026. Again, we'll be focused on our predators, our largemouth bass, sunfishes, and any channel catfish or blue catfish we can get. And these will be used for both advisories and assessments for fish consumption. We also have a fish tissue investigation this year. We learned that there is a groundwater well in eastern Kentucky in Harlan County. There's actually a sign there on this. This is pulled from Google Earth. I just pulled this from Google Earth that says, "Don't drink the water from this well." And I'm pretty sure the car there has some jugs in it for water collection. So we want to make sure that we know where the selenium is coming from, that we're determined what streams it's impacting, and to determine if our fish tissue is any of the selenium is being absorbed in fish tissue. So this year, we've already started last year, we'll be focusing on water chemistry and fish tissue in this northern Harlan County spring and the streams that feed it to determine where the selenium is basically what streams it's impacting. So finally, this is the last slide I've got. And I just wanted to give you guys an idea of what a harmful algal bloom looks like. These are the last two that we had reported and we followed up with. You can see that advisories were issued for both of them, and the date the advisories were issued and the date they were removed. Carpenter Lake in Western Kentucky near Owensboro has been a long problem for us. We've had multiple harmful algal blooms, and they've typically lasted, you can see, October to February. So we've had a hard time coming to figuring out what's going on with Carpenter Lake near Owensboro. We had a small harmful algal bloom on Harrington Lake last year that was very very seasonal. It only lasted for a few weeks. So again, these harmful algal blooms are response-based, and we're focusing on those until we can get data to demonstrate the toxins are no longer present. And those are the last two we had. With that, I know I'm just about out of time. I will wrap up, and that's all that I have. So I guess I can stop sharing my screen, and I can hand it back over to Stacy. If there's questions, I'm happy.
Yeah, so that kind of wraps everything up, but you guys welcome to ask questions. I think we have a couple from our team actually, if that's all right with you, Jacob. So I know where's my cursor? Personally was wondering, what is seining? You mentioned that earlier.
So seining is, there was a picture, there's two typically wooden bars that look like giant dowels that are in about 5 feet tall, and a net is strapped between those two bars, and it's pulled through pool habitat. Our electrofishing is not very effective at collecting pelagic species that kind of live in the water column in the pools. They typically just run away from electricity. So if we have pool habitat, we use these big seines to basically make sure we're capturing representative of what species are present. So yeah, seine is basically a big giant net that takes two people to pull.
Super cool. And if anybody wants to like raise their hand to ask a question, I can unmute you, or you can pop it in the chat, and we can get to it that way. I was also wondering, what makes an algal bloom harmful? Like, is it like, why is it toxic to people?
Good question. So we call them harmful algal blooms. They're actually cyanobacteria. And when the cyanobacteria cells lyse, they contain toxins. So basically, when you have these algal blooms, it can be very, I want to say seasonal. It can be really fluctuating depending on what's happening with that cyanotoxin, that cyanobacteria bloom. So you could go out there in June and take a sample and there not be any toxins. If there was a large cyanobacteria die-off, toxins could be released from the cells that lyse. When you have a big enough algal bloom, there's constantly cyanobacteria dying. So it's actually the toxins present within the cell wall, and there's a bunch of different toxins that can exist. We typically only get a couple in the state. They can cause flu-like symptoms. Sometimes they can cause rashes. Usually nothing major. The major issue we have with harmful algal blooms is livestock and pets. There's a lot of times where dogs might go down and drink a bunch of water out of the lake. And if they get enough toxins in them, it can cause some serious illness. So yeah, it's the toxins are coming from the cyanobacteria cell lysing is what it is.
Gotcha. And is there any one thing in particular that causes a harmful algal bloom, or is it just a variety of things?
It's a variety of things. They're typically, like a lot of things, they're nutrient based, they're nutrient driven. If you've got a low flow pond in the middle of summer and it's not getting any flow and we haven't had any rain, that we see them pop up in that. We also see them sometimes after a rain event when it's been really hot. It'll flush enough nutrients in, and then you'll see a pop of cyanobacteria that might last just a couple weeks. Sometimes they move down the Ohio River. They're very, very hard to predict. So we base ours on a response base. The public may reach out and say, I was out on the Kentucky River and I saw some weird looking green stuff, and we'll go check it out and take a sample. So that's why we have that link there on the website. Anytime we get a report of that, we'll go take a sample. If there are toxins, we'll follow up until the sample's gone, until the toxin is not present anymore.
Got it. Thanks. Can you see the chat, or would you like me to read a question that we have?
I cannot see it, but yeah, if you don't mind reading it, that'd be great.
Not at all. So Catherine was wondering, what is your team's relationship with Kentucky Watershed Watch Volunteer Monitoring Program? Do you oversee this work, or is this more the basin coordinators? And how does your team work with those coordinators?
Good question. So the volunteer monitoring is under watershed management branch, and that is basin coordinator based. We do have a member of our staff, Colin Arnold, who was managing our lakes program. Now he's actually taken over ambient surface waters as well. And he works with the volunteer monitoring. I think they have a regular meeting with volunteer monitoring. A lot of the volunteer monitoring is lakes based. As far as our relationship with basin coordinators, we typically will kind of let them know if we see issues or concerns in their basin. So if we see something we're concerned about in the salt basin, we would let the salt basin coordinator know. They may let us know about issues or concerns. If we talk to a landowner that has a question on while we're out doing field work, we often refer them to the salt basin coordinator, especially if it's something related to agriculture or any kind of non-point source disturbance. Now, Jessica Schuster's section in intensive surveys and wetlands, which is next door to my section, works with the basin coordinators often to develop 319 grant project-based, very specific watershed-based studies. So our group does more statewide and long-term monitoring, but we do have, if Colin Arnold is our staff that does work with the volunteer monitoring program some.
Very cool. Well, thank you for that information. I'll open it up. If anybody else has got questions, feel free to chime in.
Jacob, thank you so much. It sounds like your team's very busy, and it's great that you have that portal. I was curious, is there a way to see quantitative results from the information you're tracking?
Yes. So we have a database, and all of our data goes into this database. It's called KWADE.
Okay, Wade.
Of course, right now the data, I mean, of course that data, if it was to come from an outside agency, not within the cabinet, a core request could be filed. But
Okay.
yeah, we have lots of quantitative data for streams, lakes, rivers. Like I said, our ambient surface water program has been used for some separate studies that have been published because we have 20 to 30 years of regularly collected water chemistry data at a lot of our bigger rivers. So yeah, the quantitative data book can be made available if there's something particular you're interested in.
Very good. Thank you.
All right, that seems to kind of be wrapping things up. I'll see if I can
Okay.
get my screen to share one more time here, just in case anybody had some questions about ECAP or wanted to get in touch with us. Just a reminder that we have some other upcoming events in this fall for small businesses specifically, that anybody's welcome to come in. And with that, give us a call or email if you need anything. Thank you again so much, Jacob. This was great.
Yeah, thanks a lot.
Thank you, Jacob.